All right, well, we can go ahead and get started. Can everyone hear me okay? If you're a webinar attendee, that means that you cannot be on video or speak. But as we'll explain, you'll be able to participate in your own way. But yeah, let's go ahead and kick it off.
Okay, next slide. Perfect.
All right, so just a few housekeeping notes before we get started. So this webinar will be recorded and made available. So if you want to share with your networks, we are going to be using the Q&A function to take questions. So again, you are not able to unmute yourself, but you can leave questions in the Q&A function, and we'll have someone who's going to be monitoring that. We likely will not be answering questions as we go. We will save them for after panelists' presentations, at which point we will have the formal Q&A portion, and a lot of your questions will be answered. At the end, we're going to have a short survey. It will come to you, it will pop up for you, and it will also be sent to you as an email. We'll give you a few minutes at the end to be able to fill out that survey, so please remember to do so. And with that, welcome to State of the Science PFAS in Fish. What we know, what we still need to know, and what this means for exposure. This webinar is sort of officially sponsored by Cape Fear River Watch, the NC State Center for Human Health and the Environment, the Duke University Superfund Research Center, and the North Carolina Coastal Federation. To introduce our facilitators and our team. So my name is Kiara Klein. I'm the program coordinator for the Community Engagement Core of the Duke University Superfund Research Center. I'm honored to introduce my team, Dr. Liz Shapiro Garza, who is the Director of Community Engagement for the Center, and Megan Hort Hughes, who is the Senior Program Coordinator for Research Translation. My co-facilitator is Katie May, co-director of the Community Engagement Core of the CHHE, Center for Human Health and the Environment, and she is joined by Lacey Brown, who is the Community Engagement Specialist of the CHHE. I'll turn it over to Liz quickly to explain briefly why we're doing this webinar, why we're doing this webinar series, why now, what our goals are for these three webinars that we're putting on, and yeah, any kind of to explain the motivation behind why we're doing these.
Hi, and welcome. It's really nice to have you all here. I think the number of people who have responded and joined us today really speaks to the relevance and importance of this particular topic, particularly here in the state of North Carolina. As we know, PFAS has been a major concern, of course, starting with the issue of Gen X in the lower Cape Fear River starting in the 2020s, early 2020s. But really, we are seeing more and more that we are experiencing PFAS contamination throughout our state. The more studies that are done, the more that we are seeing that this is not just an isolated issue on the lower Cape Fear River. It is throughout the state, and the sources and the sources of PFAS contamination and the extent to which we are being exposed in every aspect of our life continues to shock and continues to be of concern. So that's why we're here. We are the group that you see represented and is sponsoring the work today, particularly Cape Fear River Watch and the North Carolina Coastal Federation, have done so much work, really important work, exposing this issue and exposing the degree to which it really is affecting not only human health, but also our environment. And we are very particularly interested in looking at the ways in which PFAS and these forever chemicals travel through our food chains. What is really difficult for the folks who are studying this work and are trying to communicate it, especially about this issue to the general public or to set policy, is that there is so much that is unknown, and our understanding of both the degree to which we are being exposed the ways in which that may be affecting our health and affecting the environment, our understanding continuously changes as more research is being done. So we are incredibly grateful today to welcome Dr. Rainer Lohman and Dr. Jennifer Sun, who are at the forefront of asking and answering these questions of what really is in our environment, how are we being exposed, where is it coming from, and how is it affecting our health and the health of our environment. So with that, we welcome you. We are so incredibly grateful for your willingness to spend time with us and to share your understanding of this pressing issue, especially here in the state of North Carolina. Thank you so much.
I think that is my first slide. So, as was said, my name is Rainer Lohmann. I'm a professor at the University of Rhode Island, which I joined in 2004. And at the picture below, you can see how our campus looks like in summer. Right now, it's a little whiter. I got my PhD from the University of Lancaster in 1999, and that has the old world charm with a castle on the top. And now, if you can click on the picture, a lot of the work I do is directing Superfund Research Center, similar to the one in Duke, but ours at the University of Rhode Island is focused on PFAS, and it's called STEEP, Sources, Transport, Exposure, and Effects of PFAS. And our regional focus is typically on community on Cape Cod or communities on Cape Cod, even though we work across the New England region. And the STEEP Superfund Center combines research into environmental fate and transport, was led by Elsie Sunderland on what happens to PFAS once they have been used as AFFF and go through the soil into the groundwater. We had an epidemiological study focused on the risks to babies and young children from PFAS exposure. We had a toxicological project on PFAS effects, and then a detection and bioaccumulation focus, which is what I lead. And of course, we have combined with that a training mandate, data management, engaging with community, and research translation. And with that, on my next slide, a couple of things that we do. So basically, we work to understand the process from bioaccumulation from compounds being present in water, enriching into plankton, which is then taken up by small fish, which is then maybe eaten by birds or a seal, and then going up the food chain. And so this has been our map to guide us. And the only reason I put in 3M is not because of product placement, but 3M was the first company to actually sponsor a major global study on the presence of PFOS in their case in foods, and that's of foods as in wildlife. And that was the beginning of the realization that PFOS in this case was indeed omnipresent and dominated in almost all of the species of interest. And then now we look at marine birds as one example of top predators, in this case, often great shearwaters. And another project shown here in the bottom left with Asta or Dr. Asta after Michael was trying to understand the uptake of PFAS by plankton. And now, if I have one more slide. So the other thing we do is try to understand what animals are exposed to. So we use these passive sampling tools to understand the presence of different PFAS in groundwater, surface water, marine water, depending on the time. And then on the right, you see two different outputs. The top right was a collaboration with the USGS. They installed a mobile laboratory on the Cape and exposed the fish to real groundwater. And similar to other contaminated sites, concentration were very high. And then try to understand the uptake by fish, by mussels, and by our sampling tools. And the results are challenging. Maybe I'll leave it at that. The picture on the bottom right is probably most relevant to what we're discussing here today. This was a group effort led by Jonathan Petali. At that stage, he still was working in New Hampshire at the Department of Environmental Protection or Environmental Conservation. This was to look into fish advisories for PFAS and what we know about them, what the limitations are, what has been happening. And you see from that picture that several states, those highlighted in color, have taken some action to come up with advisories or restrictions or guidance on what to do with fish. This is 2 years old, so clearly things have changed, and I'm sure we'll discuss similar veins of discussion later today. So with that, I hand it over to Dr. Sun.
All right, thanks very much. It's nice to meet you all. My name is Jennifer. I'm a postdoctoral fellow in Elsie Sunderland's group. As Reiner mentioned, Elsie is also a lead investigator as part of the STEEP program working on Project One. Unfortunately, she's on sabbatical this semester and has been traveling, so I'm kind of filling in with her and happy to share a little bit more about my work and some of the work going on in our group. Have a little more of an overview of some recent results from some studies in our group. But yeah, a little more background on me. I graduated from my undergraduate at Cornell back in 2013. Did some work on water quality science and management, kind of outside of academia for a few years out in California, before kind of combining my interests in aquatic ecology and water quality as part of my PhD in Elsie's group, which I finished last year. I'm finishing up a short postdoc right now, and will actually soon be heading off to a new position. If you advance the slide a couple steps. Yeah, I'll soon be starting a position as a senior scientist with the San Francisco Estuary Institute, which is a nonprofit applied research organization out in California. But for today, I'll focus on a couple of examples of two different kind of case studies or study regions in which we've been looking at PFAS in fish in our group. Kind of use a combination of analytical measurements and computational modeling to look at the fate and transport and bioaccumulation of PFAS in these aquatic ecosystems. So one study is focused on Cape Cod if you go back, just brief overview. One study you'll see on the left is in Cape Cod, in an AFFF contaminated watershed, looking near the contaminated site and downstream. And that work was done by Heidi Pickard, a previous PhD student. And I'll be comparing that with some work that I did recently looking at aquatic food webs in the remote North Atlantic near the Faroe Islands, as you'll see here. So you could advance the slide. Okay, so one of the themes that we've been looking at in our group is characterizing bioaccumulation beyond these legacy PFAS. As you're probably familiar with, these legacy PFAS have been phased out starting in the early 2000s and have been replaced with a variety of novel compounds, but we don't have as good analytical methods to individually quantify those novel or alternative PFAS. So we've been using what we call a toolbox of analytical methods to provide complementary information about the broader universe of what PFAS we might be seeing. So as you see on the left, targeted PFAS is what we've traditionally used to measure individual concentrations of individual PFAS compounds. Moving beyond that, we can measure things like total oxidizable precursors, which gives us a magnitude of the terminal PFAS and their precursors in the sample. But moving beyond that, we can more broadly try to identify the structures of a broad range of PFAS in our compounds using non-targeted analysis, but that doesn't give us any kind of quantification of those individual compounds. So kind of complementing that, we can also use measurements like extractable organofluorine, which can give us a magnitude of kind of the bulk organofluorine or most PFAS that's present in our sample without identifying any individual compound that makes up that bulk organofluorine. So we can kind of use all of these methods together to get a better picture of what is accumulating in tissues today. And so on the right, you'll see this figure showing trends in EOF over time in pilot whales out in the remote North Atlantic. We were really interested in looking at how EOF changes over time to see how these food webs are responding to the shifts in production from the legacy to the novel PFAS. So in humans, for example, we've seen that as the legacy PFAS concentrations have decreased, the EOF concentrations have not necessarily decreased, suggesting that there's increasing accumulation of these novel PFAS. But what we saw out in these pilot wells is that EOF actually has been decreasing since about the 2010s, suggesting that overall PFAS accumulation in these more remote ecosystems is still largely driven by the legacy PFAS, which are decreasing in response to the regulatory and voluntary phase outs, which is kind of good news. If you skip to the next animation. Yeah, so when we look into the components of EOF, we did find that most of the EOF can be explained by targeted PFAS, and within that, the legacy long-chain PFAS, which suggests that a lot of these novel PFAS are not yet accumulating in these more remote ecosystems. When we look on Cape Cod, if you advance a little bit, we don't have the samples to look at long-term trends, but we actually also saw that most of the EOF that we measured in fish on Cape Cod could be largely explained by targeted PFAS as well. Most of that targeted PFAS, again, is the legacy long-chain PFAS, but through the application of these various methods, also emerged, if you skip to the next slide, was a class of novel replacement PFAS that appeared to be of increasing concern in many of our samples, and that is the short-chain perfluoroalkylsulfonamides. So sulfonamides you might be familiar with because they are precursor compounds to the terminal PFAS, so like PFOS. But as production shifted from the legacy compounds to some of these novel compounds, production shifted towards shorter chained PFAS with the idea that they might be less bioaccumulative. And that includes shifting towards shorter chain perfluoroalkylsulfonamides, like you see here, the FBSA. And when we looked at the targeted PFAS in our whale samples and the fish tissue samples, we saw that most of what we see are the long chain legacy PFAS, which you see in the orange, blue, and dark green. But we saw an increasing component of the targeted PFAS is actually made up of these short-chained perfluoroalkylsulfonamides. If you advance, you can see in the light green, they still contribute a fairly small portion of the overall PFAS we're seeing in the samples, but it is a growing proportion, which reflects the fact that these short-chain compounds are still being produced and increasing in concentration. And again, this is kind of interesting because these short-chained FASAs were produced as replacement compounds for the legacy PFAS with the thought that they would be less bioaccumulative, but that's actually not what we're seeing. If you move one more. This is some work that Heidi did measuring bioaccumulation factors out on Cape Cod. And you can see on the left bioaccumulation factors for those legacy PFAS, which show increasing bioaccumulation with chain length. And then on the right, you can see bioaccumulation factors for the FASAs. And what we see is that because these FASAs are more neutrally charged compared to the legacy compounds, in general, they are much more bioaccumulative, and they are as bioaccumulative as the compounds that they're replacing. And increasingly, studies are showing that they may be similarly toxic as well. So this is across a variety of studies, both near AFFF and more remotely out in the North Atlantic, we're seeing that this particular class of short-chain FASAs is growing in importance and bioaccumulating, and may be a particular group to focus on for future research and potential management control. Next slide. Oh, you can skip. Okay, and then I just want to mention a couple of other, I think, relevant studies. One project that I worked on during my PhD was also developing a food web bioaccumulation model for PFAS in aquatic ecosystems. This was kind of an adaptation of a screening level modeling tool that's been widely available for legacy POPs like PCBs, but we updated the model to include mechanisms that are specific to PFAS, like accumulation and binding to phospholipids and different proteins. And so this is a model that can be applied to different ecosystems to predict PFAS concentrations in fish tissues, given known concentrations in water and sediment and kind of a food web structure. Some of the key takeaways from this is that we do see very different bioaccumulation mechanisms across different PFAS structures, and that dietary uptake, including uptake from sediments, can be a very important component of PFAS accumulation in these freshwater ecosystems. So measuring PFAS concentrations in surface water is really not sufficient to really understand what we're seeing in the fish. And then one more study on Cape Cod. Heidi also did a study looking at PFAS moving away from a contaminated AFFF site. And a key takeaway from this study was that proximity to the contaminated source was really a much more important predictor of high levels of PFAS accumulation relative to trophic level, which is something that's kind of different from what we see from some other legacy contaminants. So that's just kind of a quick overview of some recent work that we've been doing. Happy to talk more about any of this.
That was great. Thank you both so much. So I'll just say to everybody online, I am a social scientist, which means a lot of what they just said sounded like a different language to me. So please do not be shy in the Q&A. No question is too big, too niche, too broad. No dumb questions. We are so lucky to have these experts here for 90 minutes, so take advantage of that. Dr. Sun, to start, we had a clarification question in the Q&A. So could you talk a little bit more about this idea that the difference by chain length as far as bioaccumulation and say it in the clearest
Right. I know I
way
kind
you
of
can.
skipped over this fairly quickly. So there are a couple different aspects of the structure of PFAS that tend to make them more bioaccumulative. So maybe if you go back one, you can see like in the structure of the PFAS, um, sorry, just yeah. You have like a functional group head. So with the sulfonamide head group, and then you also have a tail that is made up of highly fluorinated carbons. And so the longer that tail group is, um, typically the more bioaccumulative that PFAS will be. That's like kind of the part of as we see the chain lengthening, we see more bioaccumulation. But when we look at the head group, you can see on the bottom the PFBS, which is like the terminal PFAS, the ones that have been phased out. This one is charged, which also tends to make the PFAS less bioaccumulative relative to ones that are more neutrally charged, as you can see the replacement compounds above. So I think the general thought is that if we move towards shorter chained PFAS, we may see that they are less bioaccumulative and they may stay in the water, which is what we see in large part for the terminal PFAAs. So, so like for PFBS, for example, as you see here, some of the like shorter chain compounds that have historically been produced are not as bioaccumulative, but because some of these replacement compounds have other different functional groups that make them more neutrally charged, it turns out that they actually are still quite accumulative. They can still bioaccumulate.
So that sounds like it's something to do with the chemistry of it. And we can't
Yeah.
necessarily default to shorter chain length good, longer chain length bad.
Yes, I think that is kind of an overgeneralization, and it can very much depend on like other aspects of the PFAS structure. As you can see, like within the same structural group, generally shorter chain compounds will be less bioaccumulative, but that doesn't necessarily mean that in general, short chain compounds are not a concern, as we're seeing here. I think there's also other aspects to consider when we think about short chain compounds. So like they might be less bioaccumulative, but if they are produced at very high volumes and are just present in the environment and exposure is very high, you can still see high levels in the fish potentially, which is, I think, potentially a concern with some of the ultra short chain compounds. Yeah, and I think there have kind of been other mismatched expectations, perhaps, between some of the alternative PFAS that have been produced with functional groups that were thought to make them more degradable, but it turns out that they still don't really degrade under typical environmental conditions, or are not as able to biotransform as expected. So that's something we really need to follow up on still.
Okay. Thank you. Reiner, so we're really interested, like the whole impetus for these webinars is we've talked a lot in North Carolina about PFAS exposure via drinking water, but that's not the only way we might be exposed. And we know that especially in a state with beautiful rivers and beaches, a lot of people catch and eat fish. So I was wondering if we could zoom back out a little and you could speak to what has your work shown you about the way PFAS behave in the fish that people eat, perhaps in comparison to some of the other compounds we often talk about in fish, like mercury, PCBs, or dioxins?
Okay, yes, big picture question. So maybe the good news is the enrichment of mercury or PCBs in fish is much stronger than for PFAS. But unfortunately, that doesn't really absolve us from the challenge of figuring out what to do with fish consumption. I mean, in Europe, the estimation is that 76% or so of the exposure to PFAS is from from fish and seafood. In the US, I think the numbers are fairly similar. The drinking water, which of course is much easier to target, is typically accounts for 20% of an exposure, so that leaves 80%. And now you've got to figure out what to do with the rest. The concern about PFAS in fish is not unique to North Carolina. I think anywhere people have a habit, tradition, culture, economic liking for fishing, this becomes an issue. I think there are a few guidelines we can follow, and Jennifer alluded on some of them because she showed them for Cape Cod. Typically, freshwater is much more impacted than coastal or marine food. So that is solace for those that are coastally based and like to go out fishing in saltwater. It doesn't really have those that are on the freshwater side. But that would kind of my first rule of thumb. If you have a choice, go with the salty stuff. Now, if you like that, is a different question. I hand it over to Jennifer. I'm sure she has something else to add.
I think compared to maybe something like mercury that can, once it's like out in the environment, can transform and like yeah, it is. I would say the concern with mercury, I think, is a bit more widespread compared to PFAS, where we do see very high concentrations in freshwater fish, but as like the last study I showed, I think a lot of it does come from proximity to contaminated sites. So the more that there can be monitoring and measurement of different sources to and like advisory guidelines set on particular ecosystems to identify those that are more contaminated than others, I think that can be helpful, because I do think it has a lot to do with like proximity to sources.
Great. Thank you both. We'll take the question now from Virginia from NCDHHS, who asks, can you say more about the challenge of surface water concentrations not being sufficient to predict fish tissue concentrations? At DHHS, they would love to have a more cost-effective way to predict fish tissue concentrations.
Yeah, that is a great question, and I think one where I think we're still trying to figure out. I think is a challenge we will be figuring out at my next place of employment as well. I think one of the issues is that, as I said, like there's a wide range of PFAS structures. So like what Heidi has seen, for example, often we'll see higher concentrations of the shorter chain PFAS in water and more accumulation of the longer chain compounds in fish. And for a very bioaccumulative, longer chain legacy PFAS, we might not see a lot of it in the water, even though it is bioaccumulating. So you might have levels that are below detection water, but it doesn't mean that it's not there and a problem. I think particularly for benthic ecosystem where you're having PFAS that is bound to the sediments, that might also be a different profile of PFAS, whether it's something that's bound to a particle and settled or something that's coming up from groundwater. It might be a different profile from what you're getting from other surface water sources. And if you have like, benthic invertebrates that are, essentially eating the soil and it are like in the poor water there, that like consumption of what you're getting from the sediments moving up can also be quite important. So I think especially for benthic ecosystems, which can be quite prevalent in like smaller freshwater ecosystems, it can be important to look at what's in the sediment as well. But yeah, I mean, I do think for a lot of ecosystems, many of these PFAS can occur together. So, if some of the longer chain compounds you might expect to be there, even if they're not in the water, if you're measuring a high level of PFAS anyway. But yeah, I think making sure you're looking at different exposure pathways is important.
And I think to some degree, if I just want to piggyback on this, we know this from contaminated sites that things like eels or crabs or lobsters, so anything that's on the sediment side typically has much higher concentration, a much greater concern. So I think with that, in that respect, the story of PFAS mirrors what we know from other contaminants.
So, not surprisingly, I think a lot of people were surprised by that percentage that close to 80% of our PFAS intake might be from the fish we eat. And I think this is going to be a hard question to answer because it is at the crux of how we think about regulating or setting public health guidelines for fish or other types of food that might have PFAS in them. But we have a question in the Q&A about, is there a way to think about how many meals would equal 2 liters per day of PFAS contaminated water consumption, or a way to think about the two in comparison, fish to drinking water?
Before Jennifer answered the second question, let me just, I quoted the European estimation of 76% is from fish. I think the US might be a little more challenging because it feels to me that it's almost like there's a population that's coastal that typically has a lot of fish consumption. There's other populations that are a little less reliant on fish. So I think there might be more granularity within the US, but I hand it over to Jennifer for the other question.
Yeah, I mean, I think, yeah, how much fish, I think the numbers maybe like up to 80%, kind of depending on how much fish you eat and also where you are, like how close your like how contaminated your drinking water is as well, can also, play into that. Typically, it's like at health agencies, they will set like a safe consumption level that they will back calculate like a safe drinking water level and a safe fish consumption level from. So that is something that can be done and is being done at certain state levels. Whether we are able to do that more broadly, like at the federal level, I think is maybe unclear presently, but I think there are some guidelines in some states, like, New Jersey.
Yeah, so just for your context, in North Carolina right now, we have a fish consumption advisory that's based on the levels of PFOS for a chunk of the Cape Fear River, so the southernmost part of it. And that river also extends way upstream into its headwaters. And so one of the things a lot of us on this webinar are thinking about is like, it's just not practical to think that our state has the resources or the time to create advisories for every single water body, including those where we know there is PFAS contamination. So trying to think about like what are those rules of thumb, or what can we learn from experts that, like you all, that we can apply in places where we don't have advisories yet. So one thing I'm curious about, we often have rules of thumb when we talk about how to prepare fish when we're thinking about other types of contaminants. Is there anything either of you can tell us as far as where PFAS tends to accumulate in parts of the fish, and thus are there preparation techniques that might help reduce the amount you intake if you eat the fish?
Yeah. I think like Reiner mentioned that many of these like benthic organisms can often accumulate higher levels of PFAS, especially if you're sort of near a contaminated site.
What's
Um,
benthic?
oh, sorry, like things that are like crawling on the sediment and eating, yeah, essentially down in the sediment. So like crustaceans and shellfish sometimes, things like that. But also I think sometimes the smaller prey fish may also be closer to the sediments, and sometimes they're more commonly maybe eaten as whole fish. And for PFAS, we've seen that many of them tend to accumulate more in blood and certain organs like liver and kidney. So, um, more so than you generally see lower levels in muscle, and fat. So, I think making sure to remove those kinds of organs, as part of anything you're eating. Um, I think maybe more of like the subsistence fishing kind of community might more commonly eat like a whole fish. Um, but other than that, I think, yeah, rules about trophic level um, don't apply as cleanly to PFAS, I would say. So yeah, that's I think one thing that I would say.
A couple of additional thoughts. Maybe one that I think don't we said that earlier, but clearly eating fish has plenty of benefits, so we don't want to forget about those when we talk about PFAS. And we had a meeting with most New England states to discuss PFAS in fish and wildlife, and the overarching concern among all the most folks were from regulatory agencies from the region. They said just we want people to eat fish. Beneficial for your health. Don't forget this. But, of course, now comes the but. As Jennifer alluded numerous times, it would be really helpful if states can identify water bodies that are clearly impaired by PFAS contamination and put out warnings and just say as much as you can, if you want to fish here, then release. Don't eat. Go somewhere else. I think that would be the most obvious bang for the buck. Because if we really go strictly by the EPA's logic behind the 4 parts per trillion in drinking water, you cannot eat many fish. And that's not really the message we want to get out. So now it's just about minimizing harm and maximizing the other benefits of fish as a food, as tradition, whatever else it is that brings people to fish. So I think we'll keep that in mind. If we really want to have advisories, then you would have to basically advise almost across the entire state, because that's not unique to North Carolina, that's across the entire US. There's just enough background by now that most fish are not really, you shouldn't consume too many of them. So I don't think we can go by that rule, because that would turn everybody off fish. So with that, now you're just having to, make the best out of a very unfortunate situation. And of course, in North Carolina, you have pretty large regions that are much more contaminated than in other sites. So I fear North Carolina would have to cast a wider net of saying maybe minimize fish here and go coastal, go somewhere else. That would be my hope. And I think some states, and Massachusetts has clearly identified a few ponds on the Cape where they put up signs, do not fish here, because they're just the worst. Others are just normal, and that's probably fine.
Thank you both again. We want to encourage people to keep asking questions in the Q&A. In the interim, I will ask another question that for our teams, and I think for fish consumption advisories in general, poses a really sticky problem, which is what can you tell us, if anything, about the interaction effects, cumulative impacts of PFAS in combination with other chemical or non-chemical exposures?
All right, I can go. I mean, Katie already asked about what we learned from other contaminants, and it's funny because, of course, the other contaminants haven't gone away. So the fish will still have some PCBs and might have some mercury. Luckily, concern with mercury is mostly, it seems, from what I understand from Elsie Sunderland's work, confined to tuna of various levels. So that's really very different concern. So typically, that is not as big a concern. But yes, of course, we still have the other contaminants. We know they don't do us any good either. We still have the benefits of eating fish, so that doesn't go away. But yes, you have to accept that there's multiple presence, and when you avoid the fatty fish because of the PCBs, that doesn't protect you from the PFAS. So I think to some degree you also have to accept there are limits to what you can regulate, given that it becomes a very complex cocktail of compounds present right now in fish.
Yeah, I think looking at actual mixture effects of PFAS is something that people are very actively trying to work on presently. So I don't know that we have a lot of specific guidance, but many of them do have similar structures. So we might expect them to act similarly, and at least potentially have at least additive effects. And they can, affect different structural PFAS can affect like different organ functions in different ways. So I think, yeah, I think there's definitely expectation that there will be some additive synergistic type effects, but that's, I would say, a very active area of study.
Maybe an add-on for some fish and some water bodies, there of course already exist fish advisories because of but particular contamination, PCBs or mercury, whatever it might be. So to some degree, those might be useful as a guide on what to do. And of course, now I would have to add specific hotspots for mercury, for PFAS, and of course, North Carolina has its fair share of those. But some of those existing guidelines might help protect against PFAS too. So there is something already in place, and that might help.
Can you say a little bit more about that? Are you saying that existing advisories for other contaminants might inherently also be protective for PFAS based on where they are?
I think to some degree they outline sometimes specific fish or specific regions, and that might at times overlap because sometimes these fish advisories for mercury or PCBs are because of more general contamination. And in that case, they might someone already provide some guidance on what will be useful for PFAS too. That, of course, does not help with unique PFAS contaminated water bodies.
So that's helpful. And we have two kind of similarly related questions. So we know some of the point sources, some of the large point sources of PFAS contamination in North Carolina. Of course, there are other ones like land application of biosolids and old landfills. But in thinking about maybe some of these known sources, point sources of contamination, when we think about Jennifer's point of proximity to the contamination source can be a type of predictor for bioaccumulation. Do we know about the life cycle style of fish as far as like, what if they swim away from there? Can fish near a contaminated site then go contaminate other areas? And how do we think about that?
Sure. So I think compared to something like mercury, which has a much longer lifetime in fish and can really biomagnify, and like increase with age, many of the PFAS have shorter lifetimes in the fish. So maybe on the order of like months, they might start removing them from the body. So if a fish were to swim away from a contaminated site, I think it would still hold some of that contamination, but some of it would be flushed out. I think we would be less concerned about fish like bringing contamination from a contamination site elsewhere, versus just the contamination site itself. I think studies both in our group and Reiner's group have shown that PFAS, like in the sediments and groundwater, can persist for decades, like potentially centuries, and continue to be a source to that contamination site. So I think it is very much about like the location, and fish that come to the location can also fairly quickly take up PFAS as well.
Maybe I want to just add a little bit of good news, and that was hidden in Jennifer's talk. The regulation on PFOS, or the phase out by 3M in the early 2000s, has slowly but eventually trickled through to the pilot wells that where indeed concentrations are decreasing. So there is a recovery happening. It's just slow. Unfortunately, of course, this is different for contaminated sites and point sources where it's sometimes very difficult to install effective remediation or treatment, in which case the residence time in the soil typically is very, very long. So the recovery will be much, much slower. But if you were able to not only turn off the tap, but then also find some way of either keeping the PFAS in the soil or making sure they can't escape, then water bodies and fish will indeed recover, and the PFAS will eventually be flushed out to the ocean. But we're clearly not there yet at most contaminated sites.
Yeah, related to that, Danny just asked a question. So, in thinking about half-lives of these chemicals in fish, as the fish are flushing PFAS out of their bodies, does that mean they're going back in the water and then being taken up by other fish?
Hmm. Yes, but I think like the burden of PFAS that an individual fish would take and remove in its body would probably be fairly small relative to even PFAS that's like already just widely distributed in the environment. So, um, I would not think that that would be a major concern for the spread of PFAS as opposed to other, um, you know, ways that PFAS can transport in surface waters or even, through the atmosphere, things like that.
So, we have a question about, again, oversimplifying. It's just what a lot of us like to do. It's easier to communicate that way. I'm sorry. So, to Reiner's point, if we just stay away from contamination sources or contaminated bodies of water, then are we fine to eat fish every day?
Right. I don't work for the Department of Health, so keep that in mind. Every day sounds like a lot, but I understand fish consumption is for some people a daily feature, and I don't think anybody would want to say that is not a good idea. So if that's what you want to do, yes, I would try to make sure there's as much as you can. There's a variety of things involved, variety of fish or shellfish. If those can be coastal and or more shellfish, I think that would make it better. But yeah, if that's what makes you happy, I wouldn't want to interfere with that.
But in a way, that sounds like it is connected to the level of PFAS in the water. So this is where a lot of us have been struggling for what feels like years now, is like it's not a perfect one-to-one between the PFAS levels in the water and the PFAS levels in fish. It's not a perfect one-to-one from trophic level, smaller fish versus bigger fish. It sounds like whether they're benthic or not could be a good rule of thumb, but if you had to rely on just one of those indicators, are any of them the most useful? Could you use any of them? Acknowledging that all are imperfect in some way.
Okay, I'll just say one thing, which is, I guess in saying that surface water is not sufficient, I think that is true when you're kind of getting down into the like more detailed specifics of like exactly what concentrations and what profiles we're looking at. I think if you're kind of broadly trying to survey and determine if a site is near a contaminated site, often you will find contaminated surface water and sediments coexisting. So, if you're kind of surveying for surface water concentrations and find that they're very high surface water concentrations, then like that could be a reason to look into the sediment concentrations. I think Reiner can correct me if he thinks I'm wrong, but I think it seems somewhat unlikely that you would have very high PFAS burdens in sediment without also having high concentrations to some degree in the surface water. So I think kind of starting with that screening and then kind of expanding from there could be helpful. Yeah, that's one thought.
Yeah, I was going to basically say the same. I think we have enough that you can screen, prioritize, whichever word you want to use, water bodies that are clearly impaired by PFAS and others that are less so. I think part of the challenge, and I'm not sure the word confusion, or the challenge is really that most of the fish consumption advisories are based on PFOS. Maybe sometimes people look at something else. Maybe there's a long chain PFNA. But some of what Jennifer has already alluded on is that we know there's additional PFAS present in fish, and we know they're not going to do you any good either. And this is where the sediment comes into play. This is where some of the new chemicals that Heidi and Jennifer looked at, the sulfonamides, come into play. So we know there's a lot more. And that is just for the normal sites. Now, of course, North Carolina, you have a whole different line of chemistry. So that makes the prioritization just on PFOS probably insufficient. And that's, I think, where part of the challenge is. And I'm not sure I have a really good suggestion other than you just be aware that probably in your state, it's more than just PFOS that will be driving the concern.
Yeah, I
If
think
any
like
of our state folks are listening and want to add additional questions related to that, please feel free, because that is what our advisories are currently based on, is the PFOS levels.
Yeah, I mean, I think this is sort of yeah, what Reiner was mentioning is that often sites that maybe historically have been more contaminated by PFAS would also be likely to be contaminated by these novel compounds, whereas like, a pristine environment might be more likely to be contaminated by neither. But it's just in some sites, if we are seeing like PFAS concentrations come down, for example, which we are starting to do, and in some ecosystems, it might seem like we're not seeing a problem anymore. And in those sites where we like know at least there's been historical contamination, that might be like good candidates to make sure that we're looking more comprehensively.
So I think we can sort of maybe use our context clues for this, but it would be great to hear you both speak explicitly to what is, in your estimation, what are the biggest knowledge gaps related to PFAS in fish? What are the areas where a lot more research is needed?
Yeah, there are a lot of gaps. Maybe this sounds like kind of a low-hanging fruit, but as we were talking about, there are a lot of novel PFAS that are being produced, and I think there's been up till now maybe a bit more study on exposures in humans versus exposure in like wildlife and that exposure source. So like I showed a couple of examples from a couple of locations, but I think that's not necessarily like representative of everywhere. And I think better understanding like what kinds of ecosystems might be more susceptible, or even like what kind of species or systems might be more susceptible to accumulation of some of these novel compounds would be quite important. I think there are certain classes of novel PFAS that are starting to emerge, like the sulfonamides, or even other ethers, or like Gen X came up, but like really understanding which of these novel compounds are like widespread as opposed to more localized to very specific industrial sources, I think can also be helpful to understand what to focus on more when it comes to like toxicity studies and things like that. Yeah, I'll leave it there.
Yeah, I think to just add on is if you had the funds and the time, is PFOS the best PFAS for North Carolina to focus in fish? Maybe it is, maybe it is not. I'm not sure I would be able to give you an answer. I think the other part has to do with the whole social side of fish consumption. Do we know what fish are eaten by whom, how frequently? How does that break down by different subgroups? And to what degree can you actually, if you want to effectively steer behavior to maybe a slightly better way of eating certain fish, whatever. So I think that's some of the discussion we had with the New England states was also an acknowledgement that we have insufficient data, and often it's not particularly well broken down to different ethnic groups either. So there's, I think, on the receiving side, even if a state issued fish advisories, it's an advisory. So will it actually have any impact? I think that's a really important question. How to, if you want to go down that path, how do you make sure your message actually has an impact? Because otherwise, what's the point?
Yeah, we have a question related to that. In either of your work, have you seen effective strategies for communicating about contaminants in fish like PFAS? And I would just say that we've touched on it throughout the hour, but there are people who will never give up eating fish for a variety of reasons, whether it's a cheap, free source of protein, its cultural impact. Um, and so I don't want it to be that we are trying to force people to never eat fish, but to give people the most information to make informed decisions with. So, have you all seen any strategies that have seemed really effective?
Well, maybe on the positive side, if you go to restaurants, you often see indeed some warning about mercury in fish and often women of childbearing age. You might even see some color coding. So clearly some of this has trickled through and has reached consumers and they are being exposed to it and sometimes they're aware of it. So clearly the mercury case has had ramification and it is being adopted, sometimes even by fishmongers. So I think that is a pathway to follow. Now I forgot the answer to your other question, but I'll pass it on to Jennifer. Maybe I'll figure out what I want to say.
Yeah, have I seen effective communication strategies? Yeah, that's a good question. I mean, the last project that I worked on, for example, I mean, this wasn't in the US, it was in the Faroe Islands. But they have, both mercury and PFAS contamination in their whales, and it is a very important part of like their culture and tradition to eat the whales. I think there has been a lot of communication, just like from their public health agency, um, about uh yeah, both on the mercury and PFAS side, um, about like the potential health effects on, you know, children and the the actual yeah, I mean that is a smaller community, so I think it might be like a little easier to have a response to like a public health agency. But there has been significant reduction in consumption in that community. Um, in response, I think, yeah, and just like greater awareness of the actual potential health impacts. Um, yeah, otherwise, I
If I
think,
may chime in, of course, on the Faroe Islands, they have the benefit they got away from the pyruvate, but they can eat all the other marine foods. So they
right.
stay
That, yeah,
within that self-sufficiency idea, but they just shifted to less impacted marine foods.
yeah. I mean, right. I guess for a subsistence, someone who's fishing on Cape, a lot of alternatives right on Cape Cod. Um, mm, Yeah, I mean, both on the mercury side, uh, I think for a lot of other PFAS exposure sources that even just people in my like life are aware of, a lot of communication about like exposure risks has like trickled down through the media. I'm not sure, for yeah, I think a person's like willingness to trade off the health risks of exposure and kind of the importance of eating fish in their life. I think just like greater awareness of like the health impacts that trickles down from multiple sources, whether it's like the media or public health sources. Um, I think there it did out, it can place a heavier weight on that. Um, yeah, I don't know if I have it right
It would be
here.
a lot easier if we could say, and I'm making this up, fish, let's say trout. Trout are the kind of the worst. I make this up now. If it was just trout, that would be easier to steer a conversation around because you've seen shift in the market based on availability and marketing. So clearly we can suddenly tilapia is a fish, some claim and others don't. But things have changed and people go with it. And that's also because obviously as the climate changes, species abundance changes. We see that in Rhode Island. I'm sure you'll start seeing that in North Carolina. So we have to accept that things are changing anyhow. So I think to if it was if we were able to highlight one particular fish that right now carries majority of the PFAS burden relative to everybody else, what is consumed, that would be easy. But I don't think we are in that situation, which makes it a little more challenging.
There's a comment in the chat from someone who lives near the PFAS manufacturing facility in Fayetteville, North Carolina, and they have a question, or they're making a comment about another source of likely exposure that they've had for years. But what they touch on is the idea of farm raised fish and shrimp. And I was wondering if either of you could talk about what is the potential for like aquaculture and farmed fish species as perhaps a safer or better alternative? What would make that a viable alternative?
I,
So there's a history already that we know of from farmed salmon that initially a lot of the aquaculture salmon was fairly heavily contaminated by PCBs and mercury because of the way the food was their food was introduced. Because of concerns, that was adjusted and changed, and now farmed salmon are fairly free of contaminants. I think the same will hold for PFAS because, as we discussed, yes, there's general contamination, but that's low. So I think that would be much easier to, from a PFAS perspective, yes, farmed salmon is fine. Well, having said that, there isn't that much wild salmon anyhow, so it's kind of, we don't have a choice there. Shrimp should be fine. I mean, there's concern with aquaculture, but I don't see that as a particular PFAS problem. And yes, it would typically avoid the problem of having something grown in a very contaminated area. So yes, from a Fayetteville perspective, yes. Unfortunately, of course, that takes away the fun of fishing, or you have to do catch and release or something.
There was a
yeah.
very specific, sorry, Jennifer, did you have,
Oh,
go ahead. You were
no,
saying-
I guess just to his point, just like remembering that these fish can accumulate, like the uptake can be fairly quick. So I think within a couple weeks, again, like a new fish at a contaminated site will quickly take up, um, well, what is in the water. So it really just depends on where the farm is. But that's actually Elsie is doing a number of studies on aquaculture. So you might see some more work on that coming out in the next couple of years.
Great. There was a pretty specific question about how does the bioaccumulation of fluoroethers compare to the other groups of PFAS?
I think a lot of these fluoroethers have also been increasingly observed in a wide range of ecosystems, but like for Gen X, for example, like I think some of them can be, again, very specifically tied to the location where they're being produced. I think from what I've seen outside of the very highly contaminated areas, some of them are widespread and accumulating, but generally still at like fairly low levels compared to kind of the legacy PFAS, unless we're talking about at like right at the contaminated sites. But a lot of them are not necessarily like breaking down or transforming and are therefore some of the longer ones may be just as accumulative as some of the legacy PFAS as well. But yeah, again, I think away from the contaminated sites, they can accumulate, but still they get relatively low levels.
We worked on birds, not fish, but a lot of those fish eat birds, so that might be the link. And somewhere from the North Carolina region or the Cape Fear region, and they certainly had a wide smorgasbord of PFAS, including some of the unique chemistries for the Chemours production. So some of these ethers and unique products bioaccumulate, unfortunately. Now, I think that was just a message in the question that suggests PFOS is still driving everything, even in North Carolina. So that I don't know, maybe that's good. I'm not sure if that's good, but that might be good.
All right. Well, I think as we've done a good job of getting to almost all the questions we've been asked. So in thinking as we start to wrap up, I'm always curious. If you were both royalty for a day, all the power, all the money, all the resources you could want at your disposal, what would be the first things you would do to tackle this issue of PFAS contamination in the fish that people eat? Could be research, could be policy. What would you wave your magic wand and do? You both have very good thinking faces.
This is just one. I mean, this is maybe not as specific just to the North Carolina issue, but I think from a policy perspective, and this is related to the fact that, contaminated sites are the major predictors of the high PFAS concentrations. I think like being able to simply ban or strongly restrict the production of new PFAS, except for very essential uses, would be like the first step, just because I think there continues to be new PFAS that are produced, as we've seen, that are, yeah, I think that will take a long time for us to necessarily catch up on the measurement and monitoring and like understanding of health impacts. So that, yeah.
So stop the use
very high
of the
level.
production of them first.
Yeah, I think would be very much the first
Is
step.
is that state by state? What's your vision?
I think in an ideal world, this could come from a federal place, but I think like with the legacy PFAS, a lot of the reduction came before like formal regulation. So I think there's an awareness among like 3M and some companies that they might be producing new PFAS, they might be regulated in the future, and that might cause another problem for them. I think in other places like Europe, for example, they're thinking about putting in much stronger restrictions. And for states, for example, like California, that have a lot of market power, they've been able to like lead restrictions on certain other chemicals. So I think, yeah, starting from either like the state level or like industry partnerships are all different mechanisms for trying to do that. Yeah.
Great. All right, King Reiner.
I've seen that Queen Jennifer already waved her wand, so she's taking care of the source reduction, which is nice because then I can focus on something. I would have said something similar, I think. But if if either this is already done or is not attainable. And I think states actually have a lot of power in making sure that PFAS are used in really only products where there is an obvious need at this point. And a lot of states have taken that leadership role. And I think the other next step for me would then be, if we stick to the fish theme, to make sure that generally all states are in a position where they can identify hotspots of water bodies that are strongly impacted by PFAS. And I think in some states we have seen that action and they have an idea, and others there's very little. So I think I would make sure that at least in every state, people are in a position to know whether lake or pond or river XYZ there's a concern or not.
Okay, I mean, listen, if I could wave my wand and make you both co-monarchs, I definitely would. Um, so to wrap up the question portion, unless if there are any attendees that have burning questions, now is your chance to put them in Q&A. Otherwise, I will ask you both, Reiner and Jennifer, what would you say is the most or one of the most important things for folks to take away from this webinar related to PFAS and fish generally?
It sounds like I have to go. All right, I think we have to accept PFAS are in fish, full stop. There's a lot of benefits to eating fish, but as much as possible, it would be good to identify either less impaired water bodies and fish, or sometimes different kinds of fish, or maybe fish from coastal areas, or even farm raised that are probably often better alternatives than if it's a locally impacted water body.
Yeah. Yeah, I think it was similar. Lee said, I think this is like a theme we've been talking about, just that the contaminated sites are really the strongest predictor of where we're going to see high PFAS. So really looking for, yeah, other places. And I think the other theme that also Rainer has been repeating is just that we are shifting towards new PFAS production. So looking beyond PFAS, which doesn't necessarily have to mean comprehensively monitoring all PFAS everywhere, but being able to screen for a broader range of PFAS to make sure that we're covering what's in the fish, I think will be important.
Great. Thank you both. Lacey, if you wouldn't mind putting back up our slide deck, we'll begin kind of the formal wrap up. I did misspeak at the outset. You will not get the link to the survey while you're still in the webinar. It will pop up in your browser after the webinar ends. But please do take a few minutes to fill out this survey. It's really helpful for us in moving forward and doing not only these next two webinars that we have planned, but just generally understanding what folks are gaining from these sessions. So that being said, and Katie says it's really short, I promise, and she is correct. That being said, we do have two more webinars coming up, and the next one in our series is entitled State of the Science, PFAS in Local Foods, Understanding Exposure from What We Grow, Hunt, and Harvest. So this one's going to be quite fascinating, I think. There's going to be discussion of science of how PFAS move into and through the foods that people are growing, catching, harvesting in their backyards. So chickens, backyard chickens, honey, etc. Our featured speakers are these two lovely folks down at the bottom, Dr. Li Peng Feng, who's an associate professor of OBGYN at Duke, and Dr. Ping Ping Meng, who's an assistant professor of chemistry at East Carolina. Date and time for that are here in this yellow circle, Tuesday, March 17th. That is St. Patrick's Day, but hopefully you can take an hour and a half out of your schedules to come join us to have this next in our webinar series. If we could click to the next one. Okay, thank you. Thank you. is the next thing that we want to say. So thank you so much to Dr. Sun and to Dr. Lohman for joining us. It was really amazing to hear from you both. Your research and expertise is so impressive and so valuable. Hopefully folks got to ask all the questions that they have. If not, and you have any lingering burning questions, you can follow up with myself or Lacey. Both of our emails are here. We will also send out information if you would like, not just on this next webinar, but on our third webinar as well, which we do already have a date for. That is April 21st. And so we can share that info out. Liz, Lacey, Katie, Megan, are there any things that I'm missing?
Nope. Thanks, everyone.
Thank you.
Thank you. All good here.
Okay, thank you all so much. And again, that survey will pop up. It will also be emailed to you. Please take it. It'll just take a moment.